Hello everyone, welcome to the podcast, the pIBD Digest, with me, Tracy Coelho.
And me, Chris Bakewell.
So Chris, I think today's episode is going to be one of your all-time favorites, at least when it comes to all the ohms and omics in IBD. So why don't you set the stage with today's topic, and I'll do the honors of introducing our guest of the day.
Yeah, thanks, Tracy. I would love to. So the ohm we're going to be focusing on today would be the microbiome. It's a topic that may well pave the way for the future of gastroenterology and certainly many other chronic diseases. It's much talked about in popular culture, on social media, and in the literature as well. But today is about understanding some of those core principles, which we know to be true, or certainly believe to be true about the microbiome, and thinking specifically about its role within IBD and in children. And if we've got time, we can think about touching on the landscapes of therapeutics within IBD, treating and targeting the microbiome. But given how vast that topic is, we're probably going to need to make this a double header, and we can talk about that later on.
So very excitingly, today's guest is a very special one. In fact, in the world of microbiome-related IBDology, he's basically Zeus, with a lab coat and a colonoscope instead of a thunderbolt. So now calling him a poo enthusiast would honestly be a massive undersell. As pediatric gastroenterologists, we are all enthusiastic about poo. It's kind of our thing. But this guy, he takes it to a whole new level. In fact, I was planning to rebrand our podcast for today in his honor from the pIBD Digest to the pooIBD Digest. What do you think?
It mightn't do too much for the reputation of the podcast or indeed the guest we have on today. So I'll leave it up to you. Your name appears in bold on the thumbnail, so you can fill your boots, so to speak.
So let's not jeopardize our podcast reputation. So welcoming a true microbiome maestro, a pediatric gastroenterologist, a microbiome guru, and a guitarist. Introducing Dr. Richard Hansen, complete with his own original intro music written and performed by him. So here we go. Very warm welcome, Richard.
Tracy, you're too kind. I have to say I didn't write any of that introduction, and I didn't even offer a bribe. So thanks for that. My slight Norwegian heritage would probably go more for the Norse gods, but that's okay. We can live with Zeus, at least for today anyway.
Yeah, sure. So Richard, it's becoming almost a sacred tradition on this podcast. I'm not sure if you had a chance to listen to the previous one. So we welcome our guests with well-deserved verbal confetti of praise. And I'm sure you deserve all that. So I hope I've done justice to that ritual without actually triggering dysbiosis to my compliments.
Too kind. No, I'm fine. I'm Scottish, obviously. I'm sure you can all hear that. I'll try my very best to do a received pronunciation form of Scots, but from time to time I'll slip into incomprehensible, and the consonants will disappear and it will just be a slur of vowels. So I recommend people listen back at half speed if they're struggling with that. But part of being Scottish is a sense of humility and feeling a bit like a fraud from time to time. So I do appreciate your words, but I'm confident I don't deserve any of them.
It's important to stay authentic. I think, give us the real Scottish personality.
That's it.
Richard, it's a pleasure to have you on the podcast. One of the things we like to do before delving into the proper science of the podcast is to ask our guests to introduce themselves, talk a bit about who they are, where they work, and what interests they have, either professional or if you want to delve into anything, please go ahead.
So thanks, Chris. So yeah, I'm a pediatric gastroenterologist by training. I'm currently what's called a reader in child health and in paediatric gastroenterology. Reader is a slightly old-fashioned British term for not quite a professor yet. And as you've mentioned, my main interest is the gut microbiome, probably specifically IBD, and perhaps even more than that, specifically about microbial therapeutics and IBD, and how we really move away from the current treatment paradigm of immunosuppressants and towards something that is more targeted towards what might be the causes of these diseases. I think that's going to be the best way to unlock all of this. I've been a consultant practicing in the UK for about 11 years, 9 of which were in the busy service in Glasgow as a full-time NHS consultant, and I moved to the 50-50 clinical academic post 2 years ago to set up a gut microbiome lab at the University of Dundee. So that's what I've been keeping myself busy with more recently.
Richard, I was trolling your X profile or X Twitter profile. So additionally, you are not just a microbiome maestro, you are also a very talented guitarist. Of course, impressively Scottish, according to your X profile. And I came across a legendary tale of you playing Flower of Scotland in a full kilt regalia. And that too from the top of a Glasgow shipping crane to open a medical conference. That's very impressive, heroic, and national pride. Do you want to tell us a bit more about that?
Good place to start. It's as good a place as any. So were either of you there when that happened, or is this?
No, I've seen the video, but I was gutted that I didn't go to that conference now. I feel like it set a nice tone.
So while I was working at Glasgow, myself and a friend and colleague, Professor Richard Russell, who I'm sure you'll have on this podcast at some point in the future, who's now in Edinburgh, we co-hosted he was the lead host, I was a kind of supporting host, the ESPGHAN, European Society of Paediatric Gastroenterology, Hepatology and Nutrition conference in Glasgow in 2019. We brought over 4,000 delegates to the city from 97 different countries and raised something like 3 million, can't remember if it's pounds or euros, but it was quite a lot for the local economy, which I think was a really great thing. It was great for ESPGHAN to be in our city. It was great to invite so many of our friends and colleagues to the country. And one of the key things that the organisers are charged with is putting on an opening ceremony that reflects the local culture. And I had this slightly mad idea. There's no video here, but the two hosts can see a video of me, and there's a poster behind me, which is actually for a Brian May tour. Brian May being the guitarist from Queen, who I've been a fan of for a long, long time. And you might remember that in the late Queen's Jubilee, Brian May played "God Save the Queen" from the roof of Buckingham Palace. And although I'm not at all a monarchist, that I'm not really a fan of the monarchy, to be honest with you, other than the musical variety.
So, did he get the idea from you?
He didn't get it from me. I got it from him. I stole it. I thought if we could do that but make it Scottish, what would it look like? And I thought, well, instead of a palace, what's a Glasgow institution? And we thought a shipping crane, one of these big cranes for lifting bits of boats and putting them together. And instead of God Save the Queen, what about Flower of Scotland? And so we managed to get me, it was a weird set of conversations with people to say, we'd like to do this. And actually, one of those things where you think people are going to say no, and then they go, yeah, okay, yeah, you could do that. So I found myself up a shipping crane. I'm not brilliant with heights, actually, but I was up a shipping crane with a kilt on, with my guitar, playing Flower of Scotland. And then, and we filmed all that, and we showed that in the opening ceremony. And then somebody had the bright idea, this definitely wasn't me actually, that I should then come on the stage and play it live. So I then had the dubious honour of making 4,000 people listen to me play guitar on stage. So I came on and played the second refrain of that, slightly higher up the neck, to the audience. And yeah, maybe it's one of the things that will mark who I was and what my career was. Maybe people will remember it. Maybe people have already forgotten about it, or trying or going through therapy to try and erase it from their consciousness. But it was just a bit of fun, and I think it marked out what I think Scotland is good at, which is we're not always that serious. We're quite we're quite jovial, we're quite relaxed about things, but we know how to put on a show.
Mm. Well, Southampton is hosting ESPGHAN in 2027, so you got my brain going then. I think the equivalent would be Tracy reading a poem from the top of a cruise ship. So I think that's probably nailed on for the opening ceremony for that.
Yeah, that's it. Needs
That
to be
sounds
done.
Good. Yeah. So, Richard, you have kindly shared a video clip of your performance at the ESPGHAN conference in Glasgow.
2019, yeah.
Yeah. So we'll be playing that at the end of today's episode.
Okay. It's make people go through it all again. They get flashbacks.
So, listeners, don't even think about skipping out early. And of course, right after that, we'll close things out with our traditional poetic ritual. So, stay tuned till the end. So, Richard, before we get all scientific, tell us what drew you into this mysterious world of the microbiome in IBD. Was it an instant spark, a full-blown researchy omic crush, or an omic obsession that has spiraled out of control?
Well, so it's interesting the way you asked the question, because I suppose the thing that strikes me most about that, Tracy, is that when I fell into this field, omics didn't really exist, and maybe we'll talk a little bit about that. I was an old-fashioned trainee, so it used to be that you would do senior house officer for a couple of years, get your membership, and then you'd kind of hang around for a while and do other things until you got what was called a specialist registrar post, which was 5 years towards becoming a consultant. And they called that gap in the middle the lost years, or the lost tribe. And I was kind of one of those, and I wanted to be a pediatrician, and I had a job in pediatric gastroenterology as a kind of middle grade doctor, but not as a full registrar. And it was we're probably talking about 2004, so we're talking about 20 years ago. And this was, I think, the heart of when people were excited and interested in probiotics, and that probiotics were going to change things. And I'm sure that later on, or in the next episode, we'll talk about probiotics and maybe why they haven't changed things and where that might go. And it felt quite interesting. And there was a lot of excitement around, I think, necrotizing enterocolitis and starting to understand what was going on there from a microbial perspective. And I was kind of looking at job options and everything. And this post came up in Aberdeen, at the University of Aberdeen, which was a lecturer post, which involved research. And I thought to myself that, yeah, that might be quite interesting. So I approached the people who were involved in that. And I suppose the two really important figures there are a chap called Professor Imad El Omar, who now works in Sydney, and he's the editor-in-chief of Gut, and now professor, but she wasn't a professor then, Georgina Hold, who also works in Sydney, and who's a great friend and collaborator. Even now, we actually put on a meeting in Edinburgh just a few weeks ago about microbiome. And I went and met them, and they had a lab, and they were interested in it wasn't called microbiome then. Probably we're talking about the microbiota, and people were still using words like gut flora, which is a little clumsy, and I don't think people would use that much anymore. And the techniques were not what they are now. So the first projects I did within that sphere involved growing. So you would get your DNA, and then you would either do what was called a TGGE or a DGGE. It doesn't really matter what they are, but you would run it on a gel, which would break all the DNA up in a consistent way, and you could look at patterns. And the other thing we would do was called cloning and sequencing, where you would get a bit of DNA and put that into E. coli cells, and it would break a gene in the E. coli. And when they grew white, you knew it had taken the DNA, and when it grew blue, you knew it didn't. And you could laboriously culture all those E. coli cells to find out what was going on in your original sample. But I was kind of lucky because I learned all those techniques, and I did a lot of culture-related work as well to grow target organisms, which is something I'm still very passionate about and want to do going forward, actually. But I was mid-PhD with a whole cohort of pediatric DNA samples in the freezer, and next-generation sequencing pretty much arrived. And I said, why don't I do this instead of all this E. coli stuff? And I asked, I didn't ask, this is not how it worked, but I'm telling a story. So I wrote a grant application to our good friends in Kickra, Crohn's and Childhood Research Association, who gave me, I think, £35,000 or thereabouts to do that. And we did it. And we did a next generation sequencing project, and we were one of the first. I actually thought for a long time I was the first, but I wasn't. It was Richard Kellermeyer in the States, who's a good guy. Beat me to the post over this very slightly, about So the two of us together were really the first people to publish on new onset IBD microbiomes in the literature. And I did that from Cochrane, from that PhD project, and from that springboard. So that was kind of how I got my start. The reason we were interested in new onset IBD was because we thought there was an answer there. We really thought that there was going to be an organism that triggered this whole process. And in fact, we thought that was going to be a Helicobacter. We thought that it was not my Nobel Prize. It wouldn't have been my Nobel Prize, but I would have gone to the ceremony. But the Nobel Prize that would have come out of that work, it would have been, we have grown a new Helicobacter. And I sort of cheekily called it Helicobacter coli, that causes colitis and causes IBD. And if we could just find it at the beginning, it would transform IBD the way that H. pylori transformed the stomach. So my PhD was about looking for that, and we didn't find it. And I don't think it exists. And one of the things we can talk about, if you like, as you go forward, is which organism, is there an organism? Are there groups of organisms? Which organisms? What we're talking about there? And I think the message really after all these years that it's more complicated than just a single organism. But it was a neat story. And I suppose, sorry, I've gone on for ages and I don't mean to just talk at you, but the reason, we didn't just invent that idea. The reason for that was that Helicobacters exist in two different kind of clades. There are gastric Helicobacter, of which pylori is probably the one you know most about. And actually most mammals have got their own gastric Helicobacter, including things like dolphins, which is mad to think about. But there are also enterohepatic Helicobacters that can cause colitis or hepatitis in rodents and in primates. And in fact, top tamarin monkeys in captivity get a colitis that looks just like ulcerative colitis, which is caused by a Helicobacter. So that was the justification for that. We didn't just think, here's a mad idea. We had a good rationale for it, but it didn't really prove to be true. That maybe has answered your question, maybe answered 7 different questions that you didn't even ask.
I think it's a really interesting answer. Obviously, it's really interesting to learn how you got into such a topic. I think what it says is, it's an incredibly scientific topic for a clinician to be involved in. You are the true scientist clinician. But the strange thing I always find with the topic of the microbiome is how it's both incredibly scientific and incredibly pseudoscientific. It kind of meets both criteria. In popular culture, it's, the term microbiome is thrown about, willy-nilly, often without much explanation. And I find often in quite a lazy way to explain things that are incredibly complicated in an oversimplified way. I guess the place to start would be, given that we've got quite a diverse listenership, when we say the microbiome and we say the words like dysbiosis, what does that actually mean? Because it's changed a bit over time.
Yeah, so I'll probably step back slightly from that, which is to say microbiota is a formally different term to microbiome, and microbiota defines the organisms that live within your body and call your body home, that you are their ecosystem, right? And that includes bacteria, fungi, viruses. It might include things like helminths if they're part of your environment. Microbiome takes that a step further, and it involves those organisms and their sort of activity, their interaction with you, that sort of not just what they are but also what they do. Okay, and microbiome as a term has basically started to take over, and I think to me that's okay. Actually, I think language is a progressive thing and it changes with time, and I think people like me who are maybe a little bit old-fashioned about this will recognize that distinction and will make a point of that. And I don't like when people use them interchangeably and jump between them. I don't mind if people say microbiome and they mean microbiota. I'm not pedantic enough to bother about that. And I don't bash people on the head for not using the term microbiota. I think we're heading into a phase where microbiota disappears and microbiome becomes the phrase. It's semantics, doesn't matter. But there will be people who will listen to this who will be like, oh, but I think you'll find that microbiota, they're going to write a letter into the complaints department. Microbiota is a different term. I get it. No PO box, right? So
it's fine. We don't have a PO box. It's okay.
At Tracy Coelho, right? So that's that microbiome. So you could just think of them as the organisms that live with you, but they're not passive. They're not just along for the ride. Okay. And so what I say when I'm kind of lecturing on this is there's kind of 3 things to know about this. Number one, it's individual to you. So if I look at your microbiome, it will be as unique as a fingerprint. It will define you, or we can define you by your microbiome. I've not yet seen an episode of CSI where they prove who the culprit was by their microbiome, but I'm sure it would be a good episode. If anyone wants me to write a story, I can do that. And it's functionally important. So it does things for you as a human that you can't do without it. And that's really important. Things like train your immune system, things like break down particular parts of your diet that you can't do. Number 3, this is where the gold is at the end of the rainbow. It's modifiable. We can change it. So I can't really change your genetics. If you found out that you had a gene for Huntington's disease, that would be dreadful news for you. And you would know that was going to be part of your future and what that was going to look like, and people might give you support and everything. I'm suddenly realizing I don't know enough about Huntington's disease to use this analogy, so apologies to anyone who knows more about that than me. But I can't go in and change your genetics and say you don't have Huntington's disease. But if I found that you had organisms that predisposed you towards a particular thing, or a lack of organisms that meant that you didn't have a particular function, theoretically at least, I could do something about that. I could change those. I could flip your community structure. I could move it around. I could give you organisms, or I could take organisms away. And that, to me, is microbial therapeutics, which is what the whole thing is about, And I suppose if I had a wish for my own career, it would be to transition away from being a clinical luminal pediatric gastroenterologist towards being a microbial therapeutic practitioner, whatever that is. But it doesn't really exist yet. So anyway, you asked me to define dysbiosis as well, so I will. I didn't forget. I just talked too much. So dysbiosis it's probably a bit old-fashioned as well. The way I think about dysbiosis is a review article from, I think it's June Round and Sarkis Mazmanian, who were at Caltech at the time, about 2009. I think it was Nature Reviews Immunology. And they've got a seesaw, and that's how I think about it. And basically this idea of an imbalance. So in a normal microbiome, you should have balance between good guy bacteria or girls. They actually don't have a sex, they're bacteria, right? By definition, they don't have a sex, so I don't have to worry about that. Good bacteria and bad bacteria. And usually everybody's got some of those, and they're in a balance. And in the concept of dysbiosis, at least with regards to IBD, that balance is tipped towards bad bacteria, perhaps some pathogens, and drives inflammation. That's the idea. It's really crude, and it's probably outmoded now a little bit, but it's still a useful concept for people to get their head around when they're trying to understand.
Yeah, I was gonna ask. So, in terms of that seesaw, so you've got a group on one side, a group on the other. Are these common bugs? You could name all the ones on one side, name all the ones on the other, or is it so broad and diverse that it's very difficult to define those sides, other than sort of large-scale taxonomy? What I mean to say is there are specific species that are good, specific species that are bad, and can you say who has and hasn't got a healthy microbiome because of that?
Yes, and largely no. So there are definitely organisms that you would say, when you see that's not normal, that shouldn't really be there. So like, I was going to say Campylobacter, but that's not actually a good example, because there are Campylobacter that are resident in the mouth holes. So probably like Salmonella would be a good example. If you're seeing Salmonella in a microbiome sample, that probably shouldn't be there. That's probably always a pathogen, and you can probably say that's a bad guy. We're just going to say bad guy for the purpose of this, right? But something like E. coli is really different. And E. coli is the classic example, and I use this all the time, of how complex microbiology is when you get to it, because there are something like, I think there are about 7 different variants of E. coli that are described clinically in things that they do in terms of hemolytic uremic syndrome, urinary tract infections, so, all these different things that E. coli can do. But there are also like E. coli Nissl, which is a probiotic that's been used in ulcerative colitis in the past. And it's the same species, but it's about how that species has evolved to fulfill particular niches and what it does in that context. And perhaps even more than that, what that niche looks like at that one time might define how that organism behaves as well. So if you put an organism into a non-inflamed setting and everything's pretty chill, or even if you give it a particular amount of oxygen, or you give it a particular type of substrate, it might behave in one way. But actually, if you put it into a different environment where things are very inflamed, or there's a lot going on, or you maybe drive up or drive down oxygen tensions, or you take away or give additional substrate or different substrates, then it might behave in a different way. These are biological organisms. They are adaptive. They do different things. And so it becomes tricky And this, I suppose, is one of the ideas about what a pathobion is instead of a pathogen. A pathogen is definitely bad, and every time it's there's disease. A pathobiont can exploit that and cause problems and cause inflammation and cause disease, but it doesn't always. And in a lot of people, it's relatively inert, and that might be the example I nearly used of Campylobacter, which you can find in the mouth quite a lot. It's not usually Jejuni and coli, but other ones. That might be organisms that can exploit things. And there's quite a lot of interest. There's an Australian group that are quite interested in Campylobacter. This is always a tricky one. I call it Conchysis. I think it's officially called Concisus, which is an oral organism which has been associated with IBD, and people have shown it can be adherent and invasive and stuff like the E. coli and drive inflammation. So that might be the kind of thing that could exploit a particular issue in IBD and drive inflammation in that setting, but actually in other people it wouldn't be an issue. So that trying to define that is pretty tricky, okay? And then I suppose even on the good side, so if we use Faecalibacterium prisnitzi, which perhaps people have heard about, which is maybe the poster child for the good bacterium in Crohn's disease. My friend Harry Sokol from Paris did wonderful work on defining Faecalibacterium prisnitzi and its role in actually, if you have that, you do better after surgery than if you don't have that. It was kind of his seminal paper quite a while ago now. And he's in the process of working that up as a potential probiotic therapy. One of the surprise findings from my PhD was that at the onset of Crohn's disease in children, you have more Faecalibacterium prisnitzi, not less. And one of the fascinating things about exclusive enteronutrition, which we might talk about in a while, is when you give that, you suppress Faecalibacterium prisnitzi. So even an organism like that, which you think and is always a good guy, might not be. There might be a bit more complexity to that, So I think trying to understand that even at an individual organism level is really tricky.
Richard, just a little comment. Now, with regards to your historic work and the organism Helicobacter coli that you were alluding to, I was listening to a podcast yesterday, and what I learned was you, as a researcher, cannot name an organism, even if you have kind of unraveled it by your name, but somebody else can. So if you have discovered one, you will not be able to name that organism with your name. But I could potentially provide you a name, and I was just thinking, what sort of name? And I just coined one, Hansenella coli. How does that sound?
Hansenella coli. Well, I mean, that's very kind. I don't particularly aspire to being a bacterium, if I'm honest with you.
I was going to say, I'm not sure it's a good thing, but you'll be immortalized. But
yeah. But the rules on that have changed a little bit. The rules on that have changed a little bit. So there are some organisms that are named after people. And more recently, the organisms have changed towards being more descriptive of what the organism is. And in fact, and we can go into this if you want to, but it's pretty dull, actually. In 2021, the people who decide what organisms should be named reclassified everything. And so the names of all the phyla have changed. So, for instance, Proteobacteria are now Pseudomonodota. But the literature's not caught up with that yet. So you'll still see papers that talk with the old terminology, and some papers are starting to use the new terminology. And it's very confusing. If you're not hugely engaged with the literature, reading it all the time, it's a bit weird that everything changes its name overnight. But yeah.
This is going to sound like a really dumb question, especially given your background. But when we're talking about measuring these things, how are we actually measuring them? Because obviously you can't be going to every part of the bowel and collecting a sample. So you're presumably relying mostly on stool samples, and that in itself must create challenges in terms of interpretation of what you're getting out the other end, so to speak.
So that's a really good question, Chris. And actually, I was lecturing first year medical students on Thursday last week about this, and I have a slide that I use for them, which I'm not showing a slide here, so I can't do that, but I can discuss what it shows, which is basically a beach and an ocean. And when I'm talking about this, I usually say that it's very obvious that a beach and an ocean are different ecosystems. Now, you might find that if you survey the beach, you're going to find things that are in the ocean sometimes. So you might find jellyfish that have washed up, or crabs or whatever that sometimes live in the ocean. But you probably won't find many dolphins, and you might find some humans on the beach, and you might find some humans in the ocean, but you won't find as many humans. And that idea that these two are related to each other, and you can get a signal from one that predicts or tells you something about what's going on with the other one. And if you could only survey that one, then that would tell you something about that, and you could accept that. but it doesn't tell you everything. And actually that to me describes the ocean as the fecal stream and the beach is the mucosal surface. And I think it's really important in IBD that we try and sample and look at the mucosal surface if and when we can. And I suppose when I go back to what I did initially, we focused a lot on colonic biopsies and looking at that, and we didn't really pay too much attention to stool. However, that's not to say that stool's not important, because what stool allows you is a proxy for that, but also an opportunity to non-invasively follow that up and see what happens next and subsequently. So, for instance, it would be really hard, particularly in pediatrics, to say, right, I'm interested in exclusive enteral nutrition and I want to know what happens in the microbiome in exclusive enteral nutrition. That's an interesting question, and we've done a lot on this with my pals in Glasgow. We're going to do a biopsy of your colon at the beginning, and we're going to do a biopsy of your colon at the end. Okay, for the child, that's 2 general anaesthetics, that's 2 bowel preps, that's 2 visits to hospital. And then you say, okay, actually, we're really interested in this. We want to do this 8 times over the first 16 weeks or whatever. So every 2 weeks, guess what? You're going to come in and get bowel prep and GA, and it's not practical, it's not ethical, it's not appropriate. So you're just not going to be able to do that. And even if you're thinking about this in an adult context without the GA, maybe even doing a sort of limited sigmoidoscopy, it's still like, you're just not going to get away with that. So faeces does allow us that. It allows us that window to look again and see what's happening, what's changing. And it does give us signals that are important and that are mechanistically relevant. But it's fundamental to recognize that. And when you're reading a paper, to think, oh, this is in feces. So where's the mucosal bit? What's going on there? What does that tell us? What would that tell us if we looked at it? And to think about that when we're looking at those papers.
Like terminal ileum versus colon, right?
Yeah, so Paul Ekberg, in a paper in about 2005, before the next generation sequencing era, did a survey of the normal colon. And I think he included ileum in that and showed that there's not much difference between biopsy sites. And when people look at that again and again, there's a surprising lack of difference between biopsy sites, particularly within the colon, and probably a little bit with the ileum from a microbiome perspective compared against feces. So yes, I think if you're doing ileum and you're doing colon, that's probably important to make that distinction, but it's not as big a deal as you probably think. But feces versus biopsies is a much, much bigger issue.
I fully agree. Richard, just to pick your brains on this very common scenario in the clinic. Now, families come with stacks of very pricey microbiome tests from private labs, hoping to get answers for their child's gut problems, and they may not even have IBD, just general functional symptoms. Now, when they come to me, I personally tend to politely sidestep validating those reports, especially when they come with those very glossy charts and questionable conclusions. So what's your take on this, and how do you navigate those conversations, manage expectations, sort of without completely deflating the family's hopes?
Thank you, Tracy. I think that's a really important question. I'm really glad you asked that, actually. Because it's becoming fairly commonplace, and I suppose we're talking about people spending something between £400 and £600 of their own money to get a report which they believe will tell them something. And I suppose what I would say to any of those people, or to any clinicians who might listen to this and who want to know what to say, is I don't think those reports in 2025 are really worth anything. And I try and let people down gently and say, like, I don't really know what this means. I don't really have confidence or faith in this report to know that it's telling me something useful. And so there's a few steps about that. So there's actually a preprint that's not been published in a journal yet, which I saw at a conference recently, where some clinician scientists took a mixed poo sample and they sent it to 8 different providers in triplicate and didn't tell them, and got the results back. And the results are all over the place in terms of what that same sample, which should have the same microbiome in it, should look like. So I think there's a big issue with if you think about feces in particular, feces is like a live microbial ecosystem. So as soon as it comes out of the body, it's changing. And so you have to immediately think about controlling for that. And that might involve putting it straight into a freezer. That might involve putting it into a buffer. if you put it into a universal container at room temperature and you wait 2 days, you're in big trouble because you're just not going to get a representation of what that looked like. So there's a lot of issues. I use that as an example. There's a lot of issues about taking a sample and getting it to a lab and then going through all the process and getting the sequencing and the bioinformatics to pull the stuff together, which means there's a lot of variability there. So it's hard to know what the truth is in any one of these tests, even in good labs. And then we can't really define what healthy is right now, because different people will respond differently to different microbiomes. I kind of said that earlier on. So if I swapped microbiome with you, Tracy, you might get inflammation because you've suddenly got a Scottish microbiome and you're like, what on earth is going on with this? And your body's going, where's the haggis, right? Whereas in me, it's fine, right? And so I can't define what healthy looks like to actually say that. I definitely, and I suppose I would like to think of myself as somebody who reads the literature, knows about this stuff, thinks about it, wants to change things, right? So I'm actually a pro person. I'm very supportive of microbial therapeutics and doing something about the microbiome. I definitely can't tell people what to do with that report. I can't say, it's not possible. Even, somebody who's got a PhD in topic and reads about it all the time, thinks about it. It's not possible to read one of these reports and say, aha, you have to increase this in your diet to do this or to change that. And so when these reports make suggestions based on that, that's even more tenuous. So, last word on that, I suppose, is I was one of the kind of middling, unimportant authors on a paper in Lancet Gastroenterology and Hepatology in 2025. The lead author's Porcari, which talks about this, and it gives lots of statements in response to what commercial microbiome testing may or could look like. It gives some good recommendations as well about how to make this whole field better. But one of the key things is we're not really ready for this yet within the clinical setting.
Thank you. That's really very helpful. Now, Richard, with regards to your comment about transplanting a Scottish microbiome into my digestive system, which has sampled all the delights of street foods from Goa, Mumbai, and who knows where else, I wonder how it will cope with the Scottish microbiome.
That was a joke. I'm not advocating the transplant of Scottish microbiomes to anybody, and I suspect I'd do worse with the Mumbai microbiome than you would with the Scottish one, maybe. But yeah, maybe that could be a...
People are going to start to wonder what happens in Scotland, I think.
Yeah, we could do that as a Christmas episode. We could swap microbiomes and see what happens.
That sounds like a very good plan. So, Richard, that leads me to my next question on diversity. Now, microbiome enthusiasts like yourselves often throw around phrases like microbial diversity, alpha, beta, and so on. Could you please walk us through?
Yeah, okay. So microbial. So if you want to read a microbiome paper, there's definitely two things that you need to be conversant with, right? The first one is the idea of alpha diversity, usually represented as something called a Shannon index. Okay, there's different mathematical ways of doing this, and they all do slightly different things about which communities and richness and evenness and what's going on there and how they're distributed. But the critical point about this is your alpha diversity just measures overall diversity in your sample. Okay, when I lecture on this and I talk about this, I talk about forests because I think it's easier to people to get forests in their head than it is to think about bacteria. And so I talk about a rainforest ecosystem that's very diverse. It's got lots of plants and ferns and whatever else in it, against like a wee tree that's sitting on its own in a kind of desert environment, maybe with a couple of shrubs around it. And you can see in that picture in your head that one of those is more diverse than the other one. And all I would say about alpha diversity is that the big jungle environment, lots of organisms, is represented by a higher number than the other one. Okay, that's it. That's all you need to know about alpha diversity. And so you can look at that across people and you can look at that across groups. And so classically, alpha diversity in inflammatory bowel disease is reduced against controls. Okay, that just talks about overall diversity. It's just a crude representation of that using mathematics to describe something quite complex in a simple way. Okay. The second one is beta diversity, and it's a little bit harder to get your head around. And it actually took me a few years during my PhD of reading these things and going, what am I looking at here? Because I'm quite analytical and I like to understand things. And for a long time I didn't, right? And beta diversity is when you have an XY plot and they plot dots on it and the dots look like something and they say the dots have moved or they're shifted or these communities are different to each other. And for a long time I tried to understand what the X and Y plots meant. And the truth is, they don't really mean anything, actually. And once you can unlock that, you can understand these. What happens in a beta diversity plot is it's trying to take that whole community and turn it into an X and a Y so you can plot it on a figure. And that means that you can compare jungle A with jungle B with jungle C and see what they look like. And because they're all jungles and they're maybe next to each other, they would plot next to each other in a beta diversity plot, right? So the process is they all go through the same mathematics, and communities that are the same or similar plot close to each other. And communities that are very different to each other plot further away. That's it. The X and the Y and the numbers don't mean anything useful. What matters is where they plot in relation to each other, okay? And so if you looked at jungles, which might all plot together, and then a deciduous forest from Europe, which might plot all the way over here, and another deciduous forest plots close to that, then you can start to understand what beta diversity is. So beta diversity is quite useful at looking at one set of people or condition against condition B, but it's also very useful at looking at time points and saying, okay, what happens to that overall microbial community structure as we change something? So again, my friend and colleague, Professor Kostas Yerasimidis in Glasgow, who I really hope you get onto this podcast at some point soon, because he's a brilliant nutritionalist, passionate microbiome person as well, did a seminal piece of work which looked at what happens with exclusive enteral nutrition and used beta diversity to show that actually the microbial community structure moves away from healthy. It gets more dysbiotic, if we use that term again, despite the therapy working and being successful. And maybe we can talk about that in a wee while, but that's what beta diversity does and how that helps you. So those are the two key concepts, I think, to understand.
Perfect. Really nice explanation of a not so easy concept. So Richard, before we move on to IBD, I see that you established a microbiome laboratory last year. So how is it going, and what's your vision for the future?
Yeah, so it's been hard work, actually. We're 2 years in and we're nearly ready to get going. So it's taken a full 2 years. And some of that is that it's not that easy to find funding for setting up a lab from scratch. There are actually not really funders who say, would you like some PCR machines and some pipettes? That's not, and actually if you go for a grant, you have to go with the setup already that you can deliver the project that you ask for in a grant. And you might be able to say, by the way, I'd like some pipettes, but they kind of expect that you've already got a lab and you're up and running. So that's been a challenge. And I suppose a lot of our early journey was about trying to figure out how to fund the lab. And I suppose I'm indebted to a charity called the Archie Foundation, which is a local charity for children's health in the north of Scotland, so Aberdeen, Inverness and Dundee, who gave us a kind of startup capital fund for the lab, believing in how important that was for driving forward research in our area, and the university who matched that and also supported a technician for us. So that's got us off the ground. And then it's a case of trying to identify what equipment you want, what you're prioritizing over that, going through a process of procurement where you make sure that you're spending that money well and that you're doing that ethically and appropriately. And then also slightly dull things like identifying space and making sure that it's to the right laboratory category for doing the work that you want to do within that, which is taking a bit of time as well. I set the lab up deliberately to do both molecular and culture microbiology, because I believe that's a gap in the market right now. I think, if we go back 20 years, a lot of people were doing culture-related microbiology because that's kind of what we had, and growing organisms and looking at them. And then molecular became so powerful with the advent of next-generation sequencing that that's kind of taken over everything. And it's not that uncommon now to see what I call bolt-on microbiome science, where somebody who's not really a core microbiome person has some samples and puts it through a thing anyway and gets some results back and sticks it in a paper. But it's kind of obvious that that's what they've done because they don't really engage with it or discuss it or talk about it in the discussion or have a clear. And it's not that uncommon for people to kind of get to a punchline and say, so this is higher than this, or this organism predicts this, but to not actually take that further to say, and here's why, and here's the mechanism, or here's what, the higher level science where we say not just, oh, it's E. coli, by the way, but it's E. coli because of this protein that interacts with this thing. And that's really, really where we need to get to. The only way I think we're going to get back to that is to integrating culture again. And when we find these targets that we think are important, going back and trying to grow them and then working with them laboriously to understand what's going on and what that means. I think that's really fundamental, and I think it's a challenge within this whole area. So I'm very hopeful that as we get up and running, the lab, I don't like to call it my lab, I'm not that person, I'm not arrogant enough to do that. So our lab, actually we call it the Archie lab because that was our funder. I want us to be doing, yes, that molecular stuff, but also culture-related stuff to growing isolates and understanding what's happening with them.
It's such a big topic. Like and it's so complex that you need to give it justice and talk about it thoroughly, which is why we split today's podcast over 2 sessions. So the big question then, having had that build up, is what is the significance of the microbiome in IBD, and specifically in pediatric IBD? That's a big question, but do you have a big answer for us?
Yeah, I think it's huge, and I think we're pioneers, actually. So one of the pieces of work that I've been doing that I hope will break light this year and become a paper is with colleagues in Birmingham and Sydney. Peter Rimmer actually is an adult gastroenterologist in Birmingham who's kind of led on this work. We did a systematic review of all new onset IBD papers, and I presented early work on that in ESPGHAN last year, and we have reanalyzed the data from this. So we've pulled all the original 16S data and reanalyzed it as a single core data set, which has been quite a big and powerful thing to do, I think, and we've been quite excited with the results. But one of the key messages from that was that actually pediatric IBD, at least in the new onset microbiome space, is pretty dominant. Like most of the papers come from pediatrics. So we've got a lot to offer in terms of understanding this from our patient group, because we capture them at that early phase when they've just got the disease and they don't have other confounders, they don't have treatment effect, they don't have that longevity effect of having lived with this for a long time. And so the signals that come from our patients are probably really important. And we've talked about reduced diversity already. There's, myself and others, and Gevers is the kind of big paper in this area in new onset pediatric IBD, show that some organisms are increased and some organisms are reduced, and that they're potentially pro-inflammatory organisms increased and potentially beneficial organisms that are reduced. And that's not that surprising or uncommon now. Trying to pin that down to specifics of which organisms has probably been something we've been a bit haunted with for a long time. I increasingly don't think about it like that. I think it's about functionality. I think in IBD, there's a functional, potentially functional deficit in the microbiome, and there might be a functional pro-inflammatory surplus in the microbiome in some cases. And I suppose I would point to, I'm becoming a little bit evangelical in doing this, so apologies for that, but I genuinely believe that the most important question in all of IBD is, not all of IBD, because that's not fair to people with ulcerative colitis. I care about them too, but I'm just specifically focusing on Crohn's disease for this. The most important question in Crohn's disease is, how does exclusive enteronutrition work? And the reason for that is because you can take someone who's got horrendous, severe new onset Crohn's disease, and without giving them a whiff of immunosuppression, you can switch off their disease and heal their bowel in up to, probably not as much as, but up to 4 out of 5 people by just changing their diet, by going from a normal solid food, broad-based diet to this specialist liquid formula. And it probably doesn't matter too much about which formula you give them, actually, because people have done work on that as well. But by giving them that, you switch off their disease. And so that's fundamental. It tells you that it's not the immune system, that you don't need the immunosuppressant therapies, that there's something about the interaction. This is what I think it tells you, something about the interaction with diet and the microbiome that drives Crohn's disease. And if we could understand that, we could switch that off permanently, and we could change Crohn's disease and change what it means to have Crohn's disease. And I really believe that. I don't know that that's true if you've had Crohn's for a long time. And I think if you start to get issues like perianal Crohn's disease, stricturing Crohn's disease, more extensive disease, you've had it for a long time, maybe it evolves and gets past the point of being able to capture that and succeed with that. And of course, not everybody responds to exclusive enteronutrition, which in itself is a really important question. Are they not responding because there's genuinely a group of people who biologically don't respond to this? Or are they not responding because they've not engaged fully with the therapy? That's a really fundamental question too, which I don't know that we've fully answered yet. So I think, to answer your question, actually, I think studying pediatric microbiome is really important, really interesting. We should be doing more of that, and we're very good at that. Understanding this therapy that we use a lot more than adults in a setting where we're not engaging with drugs will really help to unlock this disease and know where we go next with that.
Yeah. So what I was going to ask, talking about, I said our first episode was on the incidence of IBD in pediatrics. From a microbiome point of view, is that where we should look for the reasons why we're seeing more and more children with inflammatory bowel disease? Is there a big smoking gun there that we are discovering or are yet to discover?
Yeah, probably. So this is the whole idea of, I think, what people are calling the exposome, which is, again, there's lots of clumsy words, aren't there? And that's one of the clumsy words that we use in IBD clinical science to just describe everything that's not your genetics or your immune system. And I think the genetics is not changing. So that the fact that IBD incidence is going up and genetics hasn't changed by itself defines the fact that there's lots of people who get IBD now who wouldn't have got it in previous decades. And why is that? There is a bit of interest in epigenetics and how your genetics might be kind of modified at some level by methylation, etc., beyond the kind of gamete level core DNA, A's and C's and T's and G's stuff. I suppose from a societal level, we have less diverse microbiomes than we historically had, and those microbiomes have shifted, and our diet has moved away from diverse fruits and vegetables towards processing. We can talk about food processing all day, I'm sure. Food processing, increasing meats, increasing convenience, increased cleanliness regarding our food. Most of us used to have Helicobacter in our stomach, and now nearly none of us do. All of those things have set the scene for the development of IBD. I call it the Humpty Dumpty conundrum, which is how something happens to someone who's at risk of IBD and they develop that. And right now in 2025, we don't know how to put the egg back together again to actually switch off IBD and put someone back to health and long-term functioning. We do know how to stick bits of the egg back together with various things like infliximab that we've talked about. But actually, there's two questions there, aren't there, with the Humpty Dumpty? Number one is, how do you stop him falling off the wall? And number two, if he does fall off the wall, can we ever find a way to put that back together again? And I think those are critical questions that we need to be thinking hard about within IBD.
Beautifully summarised. And I suppose that is then a nice place to end this episode. There's a lot to think about. We've talked through a fair bit, and I'm sure there's much, much more we could have elaborated on. Thank you so much.
So, as promised, we are about to play the audio from the guitar performance video. So, listeners, hang on tight. Richard, that's very, very impressive. Now, as it is just the audio, it doesn't fully capture the magic. But even without the visuals, your talent shines through. Really impressed. What about you, Chris?
This podcast series is rapidly becoming a tour of people that are more talented than I am. So I'm impressed and depressed in equal measure.
It's very kind of you.
Thank you, Richard, for your time. It was an absolute pleasure to have you today. So see you very soon for the next episode. Goodbye for now.
Goodbye.
And before we end the episode, it's my honor to recite a couple of verses. As always, please remember, the poem does not reflect my actual views. Probably. So the title of the poem is The Mantras of Dysbiosis. As chanted so seamlessly by the bowel-wise intellects of our times, these mantras of dysbiosis and other upsurging paradigms, tinkling in infectious microbial chimes are not too easy at all to echo through my humble poetic rhymes. With the ever-shrinking gaps in wisdom of disease and its mysterious plots, disharmony and imbalance is all it takes, they say, to connect the glaring and yet so elusive dots. Is it the chaotic carnage by the ruthless bugs of the malicious lots, or our own doing through unwise greasy delights and indulgences of other sorts? Now to my learned peers, swimming in wisdom in lofty spots, do pardon me in graciousness for my cynical yet measured and honest thoughts. Thank you. So dear wonderful listeners, hope you enjoyed it. Thank you for joining. So until the next episode, goodbye from me.
And goodbye from me. Bye.